Hello, everyone. We're very happy to welcome you to the first Biological Network seminar of 2022. Today, we're featuring a Pediatric Cell Atlas. This series are generously supported by the Chan Zuckerberg Initiative. I want to take you through a couple important points before we introduce our speakers. First of all, as a reminder, please join the HCA. You can join any of the 18 bio-networks that we're currently featuring. You can be a member of more than one. Please go to the website and select the ones that you are interested in, and you will be getting our newsletters and all communications, and we will be very grateful if you join. If you have a publication that you would like to submit for to be included as an HCA publication to gain more visibility, please email to our group. You can visit our website and navigate to the publications section, and it will explain to you what to do. Please mark your calendars for the upcoming HCA events. Our next Biological Networks seminar series will feature discussion on technology, led by our Standards and Technology Working Group. That's March 10th, a month from today. In the end of March, we're having our first HCA Africa Symposium. It's virtual. Please do tell your colleagues and please do join. Visit our homepage again for details. The registration link is there. The HCA General Meeting is coming up in June. It's going to be a hybrid meeting. We do hope to be able to meet in person in Vienna, Austria, but we will also be broadcasting. Please mark your calendars. The HCA and Developmental and Pediatrics meeting is also going to be a hybrid meeting held in, we hope to be able to hold it in Paris in November. Today's agenda is pretty packed. You can see the featured speakers. We have Donna Pacicca from Children's Mercy in Kansas City, Bruce Arno from Cincinnati Children's, Kathrin Bernt from ToppCell, UPenn, Bartha Maria Knoppers from McGill, and Deanne Taylor from CHOP. We will have 15 minutes for each speaker. Please do submit your questions via questions and function on the bottom of your screen. The speakers will try to address it in writing, but we also have to have hope to have 30 minutes at the end of the talks for open discussion. So with that, I'm going to introduce our first speaker today. And it's Dr. Donna Pacicca. She's a pediatric orthopedic surgeon at Children's Mercy Hospital and associate professor of orthopedic surgery at the University of Missouri, Kansas City School of Medicine. With expertise in sports medicine, she has a special interest in knee, ankle, and elbow injuries, limb lengthening, and molecular biology of bone formation. Donna, please welcome and go ahead and share your screen.
Thank you so much for that kind introduction. And oh boy, let me figure this out. Okay, I think, is that the correct? Are we getting the correct screen?
Yes, thank you. Go ahead.
Great. Okay. Well, I'm going to talk today about our efforts on establishing a human pediatric bone and cartilage atlas. We have no disclosures. Hold on. There we go. So bone and cartilage are dynamic tissues. Bone and cartilage are both responsible for mechanical responses as well as remodeling and turnover as their structural function. Developmentally, they're an important part of embryogenesis as well as postnatal fracture repair. And physiologically, mineral homeostasis is really a key hallmark. And bone, especially, is a complex structure comprised of both organic and inorganic materials. About 69% of bone is mineral. This is a nice electron micrograph showing collagen fiber organization in bone, and then interspersed, we can see the brightly colored hydroxyapatite or calcium phosphate crystals. So bone mechanics is something I'm really interested in as an orthopedic surgeon because we deal with a lot of fractures. So we can see this cortical bone, which is like the outer layer of bone. It's very highly organized, which increases its ultimate strength. And this is one of our favorite things in orthopedics is the stress strain curve. So basically applying a load, stress will deform and ultimately break the bone, causing strain. In pediatrics, we see lots of fractures. This is what's very interesting is bone will heal by making new bone, which is essentially not scar. It's indistinguishable from the old bone. And there's constant turnover going on in bone. And bone growth occurs at what we call the physis or the growth plate. As an example of a young patient who came about a month after their fracture was reduced, and they lost alignment, and it looks pretty terrible, but we waited a couple of months, and this is the growth and remodeling that has occurred as a result of that. So pediatrics, very forgiving, and we don't have to operate on as many of these kids, but understanding these processes can really help to inform better treatments. Another thing that's really important in pediatric population is that really you're acquiring most of your bone mass for adulthood in pediatric and childhood years, and that peak bone mass is really at the second and third decade. So if we have suboptimal lifestyle factors, kids that have multiple fractures, maybe low vitamin D, they could end up going into adulthood with a much lower bone density and be at very high risk for osteoporosis. So on the cellular level, we've used skeletal diseases and syndromes to determine function of genes in bones, and then have used cell lines in animal models to identify therapeutic targets. But both cell lines and primary cell culture take cells out of their environment. So things like mechanosensation and cell-to-cell interaction, as well as bone formation resorption, are really difficult to completely study. So we have a nice in vivo picture, electron micrograph of the osteocytes sitting within their matrix, and this is compared to a cell line out on a plate. So it looks very different in vitro. And currently musculoskeletal tissues are very underrepresented in international efforts and consortia. And mainly this is due to the difficulty of isolating cells from bone samples. And both animal-derived and adult data are highly limited in their ability to find pediatric stage bases of bone and skeleton and drive treatment. So we started this collaborating with Emily Farrow, who's my colleague here at Children's Mercy in the Genome Center. And we started off just looking at bulk genes, attempting to take out marrow elements and look at, is there a difference between cortical bone and trabecular bone, looking at patients who are walking and ambulatory patients versus non-ambulatory patients. And we could see expression of typical bone genes like collagen, RUNX2, but we weren't really understanding where these things were fitting in. We can see some endothelial markers. And then when we looked at significant differences in expression between weight bearing and non-weight bearing, as well as cortical versus trabecular, we've had some novel genes that weren't primarily associated with bone development, but it was unclear which cells were driving expression. And bone is heterogeneous, and we still saw no in bone cells. So we decided to look at probing this at a single cell level in order to uncover which genes activities were really caused by which cells. And so this is something that's obviously been used in neurologic studies, cancer research, the current cell atlases, which are really demonstrating some really unique and exciting findings. So we decided to apply this to bone. Now, how do we do this? We're faced with the barrier of getting the cells out. Musculoskeletal tissue has cells embedded in a dense matrix, and basically, sorry, I'm moving my screen around. Cartilage, tendon, and ligament all have cells embedded in type 2 and type 1 collagen, and bone has a calcified matrix on top of it. And the challenge is to dissolve the mineral in matrix without destroying the cells. So we basically wanted to use single-cell RNA-seq to identify different populations in the same subtype family, establish a reference atlas, and then to correlate this with histologic data in the setting of specific diseases that affect MSK tissues. And our hypothesis overwhelmingly was that we would identify osteoblast, osteocyte, osteoclast, and chondrocyte populations based on patterns of gene expression. And then within these populations, there would be subpopulations reflecting different functions and geography as well as tissue type. So we were able to harvest primary tissue from patients, isolate primary cells, and perform single cell isolation and sequencing. And then we also thought that maybe we could compare cells harvested from different areas, so long bones versus accessory digits versus spine and skull. So this is tissue ordinarily discarded after surgery. My colleagues throw away a lot of bones, so I take it from them. And we basically initially prepare the samples, basically manually separating the soft tissues. The soft tissues out, and then kind of cleaning out, trying to wash out as much marrow as we can by vortexing. We then have a sample here. We can see the cortical bone on the outside, the spongy or trabecular bone on the inside, and then we morselize that into small fragments. And then this undergoes several hours of digest, followed by filtering and pooling of cells from that specimen. And this process can take 6 to 8 hours, depending on the size of the specimen, and we usually get about 1 to 50 grams of tissue. It's important to know that we're actually separating out the cortical and trabecular bone and analyzing those separately. The cells are then frozen while awaiting single cell capture, and then we did try doing some cell sorting to eliminate debris and improve our live cell capture. So we then capture and sequence cells by the protocols listed, process and cluster them, and then our bioinformatics team performed a detailed analysis. So to date, we've processed over 150 samples now, ranging in age from 8 months to just under 18 years. We have both male and female patients, ambulatory and non-ambulatory, and typical cell capture that we're getting is far less than what we see in live cell yields. So our initial data set for full analysis consisted of 14 patient specimens over a broad range of age, and we isolated 41,000 cells that were clustered via Seurat, as shown. And then we applied specific sorting parameters after going through our data initially to identify cell types. You can see the sorts that we used, and our CMH bioinformatics team worked with us to generate some heat maps. And this is just looking at the cartilage population, for example. We ended up generating some heat maps of known genes, as well as some up and down regulated genes. So this was really interesting, but we were really doing this on a very limited and primitive basis. So I was looking at the data saying, wow, I can see the things in the growth plate. I can see where cells are, clusters are localizing to the proliferative zone, to the hypertrophic zone, what are the articular chondrocytes, but we needed a higher level of analysis with experienced experts. So we turned to Deanne Taylor at CHOP and Bruce Aronow at Cincinnati Children's to provide a much more sophisticated analysis. So this is what I get. And let me tell you, as an orthopedic surgeon, I'm good at what I do, but this stuff I'm like not good at. So here we're looking at the process, going through the heat map to basically to identify cell markers per tissue type and cell class. And then we started renaming the numbered clusters based on the major gene markers that were present. And so basically we did normalization, harmonization, and integration of cell types and across batches and tissue subtypes. And so basically Bruce and his team took the cell data. They were able to sort out the tissue types, and this helped identify major classes of cells without forcing harmonization of the cell types between two different compartments. Then we were able to see subclasses within the lineages and then specific cell types. And this is still a work in progress. So we ended up with 107 cell signature types, and that's showing the overall heat map, and I'll show some later organization. But we had 4 lineages: endothelial, hematologic, mesenchymal, neuronal, and that was only in cortical bone. And our mesenchymal subtypes are associated with chondrocytes, osteoblasts, and osteocytes, as we expect. But we could also distinguish between developing and mature myeloid lineages, as I will show. So this reorganized heat map represents all of the cells and is sorted out by tissue type, allowing us to compare signatures across each tissue. And while there's some overlap, we've identified some specific differences in cell types that are in different tissues. And then as you look across the map, moving across columns allows us to compare cell type to cell type, while moving down the rows allows us to compare signatures within each tissue. The endothelial signature, for example, increases in complexity, going from cartilage to trabecular bone. And the hematologic lineages are most apparent in trabecular bone, likely representing the full spectrum of all of that tissue in the marrow space, but are present in cartilage and cortical bone to a lesser degree. The myeloid lineage shows signatures that are very tissue specific, with complexity going from cartilage to trabecular to cortical bone. So looking specifically at the osteoclasts, we can see clear differences in the heat map comparing across tissues. And this is very specific signature for the cortical osteoclasts, where they're expressing both TRAP, which is ACP5, as well as RANK, MMP9, and SPP1, where these were specific to cortical bone. Now, interestingly, the vitamin D receptor is only seen in trabecular bone. So this is really an interesting finding. So now we turn to the mesenchyme, which is representing all of the osteoblast, osteocyte, and chondrocyte signatures, and we see overlapping and distinct populations with specific enrichments within tissues. We focus specifically on cortical bone, where we can see enrichment of gene expression in mesenchymal tissue. And note the top of the map is basically showing our relative contributions of tissue to each cell type mapped. And the cortical bone had 44 different cell types with 33,472 genes. Within the cortical bone tissue, there were 14 osteocyte cell signatures, and we could appreciate that the osteocyte 1 cell type outlined on the heat map here had a unique signature, including specific mesenchymal genes, but also expressed some proliferative genes that were also expressed to a different degree, a higher degree in the osteoblast group. So then we pulled out the specific set of genes within the signature and used TopGene for functional enrichment mapping. So as we map our gene signature to specific notations, we note that the GO annotations show very nonspecific ECM associations, likely due to underrepresentation of the cell type. So we're limited by the lack of available knowledge. This is kind of a recurring theme. We start to see more specific associations as we move to mouse and human phenotypes and pathways associated with limb morphology and skeletal physiology, which is kind of things I would expect as an orthopedic surgeon. But we still have limited data from PubMed searches, interactions, and transcription factor binding sites. So we start to look at this, and just by what we know from the gene signature, we think that the osteocyte 1 cluster is the most basic osteocyte type, and it appears to be very distinct from previously published cell types. So we see that this osteocyte type is associated with ossification. We wanted to explore that functionality. So we took the known genes from that cluster shown on the right and had them in one pathway, and then added the other genes from the cluster that were not associated with ossification into a functional gene regulatory network. And so we get this very complex network map, which is very colorful. And I'm really indebted to Bruce and his team for this. And this demonstrated that the genes involved in ossification regulation are appearing in the center. But there are clearly genes to the left and the right of this map that are interacting but not directly involved with ossification. And we see that the ossification genes are also associated with bone length and shape as well as bone strength. The non-ossification genes to the left are associated with signal receptor activator activity and linked to the ossification genes. And they also interact with focal adhesion, collagen, and integrin binding, as well as growth factor and calcium binding. And we can appreciate how this osteocyte type has complex interactions with matrix as well as cell signaling. So moving back to the physis, a subject here and dear to my heart as a pediatric orthopedic surgeon, we're still working on this analysis, but when we look for specific known genes like those that we identified in that primitive heat map, we can get validation of functional levels. So for example, COL10A1, Indian hedgehog, and PTH1 receptor are localizing to the same chondrocyte cluster, which also is expressing Osterix, but PTHLH or PTHRP, FGFR3 are localizing to a different cluster, which is exactly what we would expect. Interestingly, growth hormone receptors doesn't localize to chondrocytes, and we do see a chondrocyte cluster overlapping with osteoblast markers, probably indicating a transitional cell. Now in osteoblasts, and this is still underway, this analysis, we see a subtype in cortical bone, which might represent the precursor to that osteocyte 1 that we talked about earlier. And interestingly, growth hormone receptor is expressed in the osteoblast population, which was very intriguing. And we also see some disease-associated receptors important for mineralization and bone strength. So additional findings that those somatic sensory neurons that showed up in the neuronal pathway, these are similar to visceral neurons in GI and heart, and these localized only to cortical bone. It was about 215 cells, so a very small cluster. And there were 3 clusters of precursor cells, very quiescent in terms of gene activity, and these seem to be just in the trabecular tissue. So what's missing? Traditional adult markers of bone are not readily apparent. SOST is missing in action. We're not sure where these cells are localizing within the bone. Are there specific areas within the bone geographically? And are some of these precursor cells more specimen specific, like from accessory digits? So why is this all important? I'm going to show you a couple of clinical cases that I think hit this home. So this is a 10-year-old young lady who presented to our sports medicine clinic, and she was complaining of pain and grinding and popping in the elbow, as well as loss of motion and no history of injury. She had seen an outside orthopedic surgeon who had commented about closing growth plates in her elbow, and mom was very concerned as the patient is short-statured. Now, in further discussion, mom, grandmother, great-grandmother, and aunts are all short-statured. They brought me pictures of the family to show all these very short people. And mom had a genetic workup as a child for her short stature that was negative, and she had received growth hormone treatment, but was unresponsive. And she's also had some problems with knee surgery and had some loose cartilage removed. So this is looking at, and actually let me go back for a second. So when we see on the lateral view here of the elbow, there's definitely looks like a little bit ossific density that's sitting there within the joint compartment. And so when we get the MRI, we can see a loose body here. There's definitely some abnormalities in the radial head. That's not a normal cartilage bone signal, and another loose body right here. So she was taken to the OR. She underwent arthroscopy. This is one of several loose bodies that I removed. And we additionally see this is her radial head here. There's a full thickness defect in that radial head. So something is spontaneously causing this young lady to have abnormal cartilage and loss of cartilage integrity. And genome analysis actually showed a shared variation of unknown significance in aggrecan between mother and patient, which was a novel finding. Our second case. Okay. Second case is 11-year-old man who had presented after a knee injury. negative x-rays, but these clearly show some abnormalities here in that bone. And he was referred. He had an obvious loose body. Basically, the entire posterior aspect of the lateral condyle was detached, and he appeared to have a loose piece here. He had an associated meniscus tear, taken to the OR, where we can see significant abnormalities within this joint. And then subsequent to that, last year, he developed a tibia fracture and then had a spontaneous avulsion of his calcaneal apophysis. So clearly, these are areas where we can use our reference data to ask questions about bone health and healing. So our data demonstrates different genomic signatures in multiple cell types. And more importantly, cells in different tissues have distinct signatures. So this has implications for how we test these cells. And so a better alignment with and validation of in vitro and in vivo models can be helpful for this. And this sets a foundation for precision therapeutics and may help define markers for monitoring of bone and cartilage health. So our future plans include spatial genomics as well as evaluating age, sex, and mechanical differences. Again, in the OR, it takes a team with multiple talents to accomplish this work, and I'm deeply indebted to Tammy Brown, my lab associate, for work isolating cells, to Deanne Taylor and Bruce Aronow and his team for the amazing analysis, and to my partners at CMH who have graciously contributed to samples, and I'm happy to take any questions or answer stuff in the chat.
Thanks so much, Donna. So yeah, please do submit your questions in the question and answer function on the bottom right of the screen. And Donna will try and address them as our next speaker presents his work. Our next speaker is Bruce Arno. He's a chair of biomedical informatics at Cincinnati Children's. He also serves as a co-director of the Computational Medicine Center. His team is using stem cell-derived cells and organoids to study the developing kidney, lung, and brain. Bruce, go ahead and share your screen.
Thanks so much, Ellen. Wow, Donna, that was amazing. I'm going to go much slower. I have just one goal in here, which is for Cincinnati to win the Super Bowl. And so I don't have to work too hard for that. They worked really hard. Okay, let's see. I'm going to go. I need to share desktop too. Is that coming through?
Yes.
Okay. And I think I need to switch. Use slideshow. So is this the right view now?
Yes, now looks good. Go ahead.
Oh, interesting. And I'm not getting the right cursor control, so I'm gonna have to disconnect this other monitor. That could hurt.
I can see your cursor though.
I couldn't.
Oh, really?
Okay, so all right. So I'm going to tell you about ToppCell. It's a web portal that we're trying to really unify lots and lots of data sets and to allow for doing interesting things that come pretty much after data sets have been analyzed through Seurat or ScanPy and other great methods that are getting developed. But once clusters are developed and data sets are processed, how do we really share data with people such that they can use it? I mean, that's why we're generating this data, is for people to use it to understand their systems in ways that they really couldn't have without this. And the bone is just a perfect illustration of that. Part of multiple consortia that are really amazing and fun and exciting. So going that direction is great. Let me talk about a couple of things. This is what ToppCell is trying to do. It's trying to use prior knowledge to help interpret single cell data and to use everything that we know up till now about tissues and cells and genes and gene networks, regulatory mechanisms to learn more from the single cell data. And so we want to be able to compare and contrast across different samples and studies, really understand development, homeostasis, and genetic effects on disease, and set up a framework for smart machine learning across all the data in such a way that we could really have translational medicine enabled. Okay, so this is my view of biological systems. It's just the most generic idea is that we've got molecules and activities of either molecules or entities that are formed from molecules. and they develop, they interact, they adapt, and then they lead to biology. And some of the principal functions in biology involve to get complexity and be a healthy organism. We have to develop and achieve organ function and prevent oncogenesis and other sorts of genetic diseases. And in pediatrics, there's no more dynamic system than development through pediatric growth. That there's so many amazing accomplishments that are performed by biological systems to do that, and so it's really just an incredible opportunity to see dynamic effects and how we get to be healthy, and how we can help people who are not healthy be healthier. Okay, so here's a framework that I want to propagate to the way that we look at single cell data post hoc to infer systems understanding. And so here's all our entities that that are include measurable properties of single cells. We're going to try and interact those entities and biological context through everything we know about biological mechanisms, protein interactions, transcription control, allelic interaction effects, metabolic states, all processes that are critical in life. And then how does all of this result in healthy functions of tissues or phenotypes, which are really disturbed functions of tissues? When you think about disease phenotypes, what are the perturbations of physiological function, and then how are those understandable in the context of what cells are doing in an ensemble structure of a tissue, a subcompartment of a tissue, a niche, or in an overall organism level where different organs are communicating with each other to have physiology working. Okay, so here's this model of a gene-centered knowledge network. These are all different dimensions of information that we know about genes, where they come from within a chromosomal context. What are their cis elements behind them? How are they regulated by perturbants, either physiological or non-physiological, by mutation genetic effects? What are the effects of different mutations or things that interact with genes, and then what's the encoded functionalities, and how do we think of the effects of the different kinds of encoded entities and what they participate in? So way back when, we had gene ontology information that where you talk about the molecular function of the gene product, where the gene product worked in the cell, what biological process is it part of, and this continues to be fantastic work that is helping us assemble a view of biological systems, but it's not enough, and so all this the information about what happens when proteins interact, what pathways are they part of. Pathways are not particularly well represented in biological process. And then the effects of mutation of the genes, like what's the resulting phenotype that happens, and so how do you organize the phenotype? So cancer is a phenotype that frequently comes from mutating genes associated with biological process of cell cycle control or DNA integrity or chromosomal integrity, but they're not going to intersect by more than 50%, and there's going to be plenty of phenotypes that relate to things like bone development or abnormal bone development that aren't in an association of a gene to GOBP. Anyway, so we've got to use all this information to help interpret a system. Okay, so now go back to single cell data, and Donna showed you how we go from nice normalized cluster structures within data sets to then modularized representations of these data. And essentially, it's a sequential differential gene expression test, and there's different tests that you can do and different comparisons of, like, are you comparing at the lineage level to the not a non-lineage or a cell class level, subclass level. So and the question is, what's your comparison group? And so you've got multiple variables that are in play here. Really, you want to try and understand as much biology as you can about these individual cell types. But if you don't, let's say, include the fact that all these subclasses were members of a class called epithelial cell types, then you'll lose this ability to find a gene expression module that spans either all of the epithelial cells or important subgroups of those epithelial cells. And you could see some of that in the module structure that Donna was showing. And I guess the thing that I want to get across to you is that the biology of any one of these cell types that occurs within the context of a class of cells is their biology is determined by the interaction of the gene functions that are engendered within the class level gene module as well as the subclass gene module. So you have to make then a stack of these gene modules that then can express the biology, or that you can use to unravel a system. And so this is the work of Kang Jin and Eric Bartha to put this together. And I kind of want to go right to the website and show you how to interact with it, because that's the main thing that I want to get across, is how you can actually use this and interact with the data and go places that you wouldn't have thought of. So we've got ToppCell divided into different project areas, and that was Eric Bartha, who's the engineer behind this, and just spectacular programmer and thinker. And so here's this crisscross of modules that were derived from each of these different conditions, and then where you can compare them going across and see are there differences between any of these conditions. And that can go for developmental stage, by a disease perturbation, drug response, anything could be used as sort of class level tests of what's changed within the cell ensemble, the cell population that's present in these different circumstances. And here's an important bar graph down here that shows the numbers of cells of each class that are present in these different conditions. So, it's not only just the, occurrence, non-occurrence of a cell type changes. It's very important to think of the changes in numbers of cells as you go from one condition to the other, as well as their transcriptomic state that they're in. So here's just a quick example of in COVID, where we could define these gene expression modules that were apparent in normal cells that aren't particularly different in individuals with, and this is just in, I can't remember if this is blood or bronchoalveolar lavage. I think it was bronchoalveolar lavage. And then these are in severe disease, there's a bunch of just genes within these modules that are downregulated, or genes within those same modules that are upregulated. So now you've got a structure to consider the biology of each of these disease-affected cell types, where you've now got a base transcriptome that's determined by these modules up here that come from the different cell types, and now you're appending on to that the gene modules that are coming from, that are differentially activated in the disease state. So okay, so I'm going to flip to one other thing to think of now. So there's biological context, perturbation states, and now how are they affecting the whole ensemble of cells that are present? And so I want to actually show you interactively a fabulous data set that was generated by Jason Spence's group in Michigan. I think working with some UK people too, the fetal lung atlas, and that it's this is a pretty early series of fetal samples, and so we've got anything from 8 to 15 weeks of gestation in here, 3 different tissues, distal, lung, trachea, and airway, and then we've divided it into these lineage and classes, and this is just using basic markers that are useful for identifying the cell types. And so now think about what happens when you divide this out by age-specific effects. So the first is within each compartment here. So here's I can't remember which tissue this is. I'm so sorry, but I think this was distal and airway and then trachea, and so as you go down these gene expression modules, this is just for the marker genes that were useful for the class or the cell type identification. You can now see really important changes that are occurring within a tissue as a function of developmental age, or then differences also between tissues and so there are different dynamics that are going on in each of those conditions. So let me flip to how much I have left.
You only have 1 minute left, Bruce.
Oh, that's not good. Okay, so here's the website ToppCell, and so you can go to these different groups of studies, and I'm going to just go to the lung map view, and I'm going to go to the fetal sample and down here, oops, sorry. Okay, and then click on this link here, and then you get right there, and then you get a preview of these different groups of gene modules. And so let's say we go for in distal lung, we're going to go for and I'm now within the week 15 of gestation. I'm going to look at the cell types that are present in this tissue that are now within the epithelial lineage class. And so here's for example, and this is just a preview of the heat map that you would get out. So you click this to generate a heat map. But I'm just going to take a bud tip progenitor, and so I'm clicking on this gene list, and I can't make it bigger. But anyway, so here's a list of the genes in this module, and so it's mapping not just to apical, basal, epithelial annotations, but it's also involved in seeing morphogenesis and abnormal lung physiology. So here's a little button that then lets you compare that gene list to known biological functions. So I'm just going to submit that gene list, which came from this one E15 developing epithelial cell, and I'm just going to let it run against all of what we have in TopGene. And the important part of this also is that all these modules from ToppCell are now in TopGene, so you can compare against this library of modules. And so here are the properties of that gene list. So it's about epithelial development, nothing specific to lung at this level. But now if you go in the category of mouse phenotype, like I was saying, there's great insights that are provided by the genetic effects analysis. So
Bruce, 20 seconds, sorry.
All right, so down here, this is the new part of TopGene that's coming from ToppCell, where here's this gene list that I used as the probe, and 197 genes of the E15 distal epithelial bud tip progenitor, and now you can see its intersection with other data sets that are in there, and so you can see it really drops out. I mean, there's just going to a different developmental stage in that exact same data set. Now you lose 50 genes, 54 genes, and then and so on down the line, and now you start to go just one more click here. Here's a comparison now to the adult human lung cell atlas from Mark Krasnow's group, and there's only 70 genes that interact that overlap with pretty much what should have been the same cell type. This developing epithelial cell type.
Bruce, we need to go to the next speaker. But
Okay.
Can I kindly ask you to drop the link to TopGene into the chat for everyone to see? Answer any questions that people might have in the Q&A function, and we hope that if there are more questions or if there's time remaining at the end of the seminar, you'll get a chance to take us through the rest of it. Thank you so much. I'm sorry about that. And our next speaker is Dr. Kathrin Byrne. She's a pediatric oncologist at Children's Hospital of Philadelphia and an assistant professor of pediatrics at UPenn Perelman School of Medicine. Her lab is researching molecular mechanisms in leukemia. Kathrin, please share your screen.
Hey, screen hopefully should be visible. No?
Nope. Now it is. Yes,
Okay,
now
great.
it's visible. Go ahead.
So thank you very much for inviting me to share some of our early data as well as some of the challenges we have encountered in our attempts of making a childhood hematopoietic and immune atlas. As I'm sure you're all aware, there are massive changes to hematopoiesis and immunity with age, and those are actually particularly important in these first few years of lives. Babies are born really without any kind of adaptive immunity to speak of. Innate immunity is immature, red cells make fetal hemoglobins, and it is in these first few years that really all the critical changes are happening that are required to set up a healthy immune system that's functional, that's well controlled, and that will ensure a healthy life. And to a certain degree, I know I'm preaching to the choir here, but it's actually crazy to me that understanding this wasn't part of the HCA right from the outset. So these changes are really reflected in some of the very, very basic parameters already that we are looking at as clinicians. You know, we know our CBC starts out with a myeloid bias, then becomes lymphoid in childhood, and then gets a worsening myeloid bias that gets quite pronounced in the elderly and actually has been linked to a host of very diseases that affect a large proportion of the population, including hypertension, diabetes, metabolic syndrome, et cetera, et cetera. Bone marrow cellularity changes with age. I don't need to explain much. These pictures are very obvious. And actually, the bone marrow of children contains a population of cells that is really absent in adults. These are hematogones, which is an expanded bone marrow B cell precursor population that's specific in children and that can reach up to 20-25% of all cells in the marrow. In addition, and this is something that I care about as an oncologist, we know that many of the diseases that affect both hematopoiesis and immunity have strong influence of age of onset. And as an oncologist, I'm just going to show you a few examples from my practice. So here you're looking at the incidence of the acute leukemias. Myeloid leukemias are really increasing in incidence with age, versus acute lymphoid leukemias, which are really predominantly a disease of childhood. In addition to that, the kind of mutations that we see between children and adults are also very different. So here we are looking at single nucleotide variants, and you can see that children in blue really have predominantly signaling mutations in RAS/KIT, while adults have predominantly epigenetic mutations in ADNMT3A, IDH, TET2, as well as T53 mutations. In addition, a lot of AML in childhood is actually driven by gene fusions. And this is, again, a typical pediatric and young adult issue. We don't really see this that much in the older population. So really, leukemia in children is a different disease, and you can't study it by studying adult bone marrow. In addition to that, there's even within a molecularly defined leukemia, age remains a prognostic indicator. Age overall is the biggest predictor of outcome for leukemia treatment that we have. You can see that we're doing quite well in curing our pediatric population, as long as the children are over a year of age. Those that are older do poorly, and those that are younger do poorly. And that is partially because many of them have a specific rearrangement in a gene called MLL, but there's actually a molecular independent influence on age that I will get back to in a little while. So overall, I hope I have shown you that really the incidence, type, and outcome of hematopoietic malignancies that we see in children is completely different and is strongly influenced by age, and we really need to understand the underlying age differences in order to make any headway with these diseases. So that was really the main impetus for me to get involved with our what we call the CHEDA, the Childhood Hematopoiesis and Immune Development Atlas, that is funded by the Kathrin Bernt Foundation. So the main reasons for this was that, as I hope I just showed you, the hematopoietic and immune development undergoes massive changes in the first years of life. And these really cannot be inferred from studying adult tissues. In addition, there are many genetic, immunologic, and hematopoietic diseases that manifest in childhood. I've shown you the hematologic malignancies, but this is actually a much larger issue. In addition, the age of onset is a critical determinant of incidence, type, and outcome. Again, I've just shown you the data for the hematopoietic malignancies, but this is again true for a much broader group of diseases. And just generally, setting up a well-functioning and balanced immune system is indispensable for a healthy life. In addition to affecting, classic immunological or hematologic diseases such as infection, hematologic malignancies, immunodeficiencies, atopy and asthma, and autoimmune disease, there's also a strong influence of the immune system onto metabolic syndrome, cardiovascular disease, really big problems on an epidemiologic level, as well as probably in all cancers through mechanisms of immune surveillance. So what are we planning to do? So we have assembled a group that spans CHOP and Penn to try to sample the entire hematopoietic and immune system. So we're starting out with the bone marrow, which contains our stem cells, B cells, and a lot of myeloid development. We will be sampling tonsils to give us an idea of B lymphoid development and mucosal immunology. We will get thymus to look at T cell development, and then we will look at both blood and lymph fluid for our mature populations. So one of the key aspects of doing this that I think probably everyone here, maybe with the exception of Donna, who gets lots of bones from her collaborators, is struggling with is how to get healthy tissue from truly healthy children. And so for bone marrow, what we decided to do is to rely on healthy matched SIB donors for hematopoietic stem cell transplantation. So for reasons that we can discuss if there's interest and time later on, we like to go to the iliac bone for sibling donors and actually get true bone marrow, not peripheral mobilized stem cells. And then because we don't want to transplant and infuse any kind of little bone chips, these go through a series of filters, and those typically get thrown into the trash, except here we get them and we flush them and we cryopreserve any bone marrow that was left over and trapped in there. So the advantages of these sources, those are truly healthy children. They have been characterized up to the zoo. And the specimen quality is really excellent because these are specimens that are designed for transplant. Challenges are a little bit of complex logistics. And then infants are really underrepresented, because there's a shorter time window to catch them. And then oftentimes there's a cord blood available, so that makes it hard to justify a major procedure. Cell numbers can sometimes be limiting. And then we don't really have an abundance of donors, so building a gender and racially balanced cohort may be difficult. On the other hand, for our lymphoid tissues, getting a truly healthy donor has been a challenge because there's not really a good reason to remove a healthy lymph node, and when they're healthy, they tend to be tiny. So what we have resorted to is getting tonsils. In a way that's nice because we get both mucosal immunology represented as well as sort of more classic germinal centers that are very similar, if not identical, to those found in lymph nodes. And we get them mostly from children who get recurrent strep throat because these kids typically at some point get their tonsils out, but not when they're inflamed and infected, but in the interval when they're healthy. Some of them are also from children with tonsillar hyperplasia that leads to snoring. Advantages are these are quite accessible and abundant. These are very common surgeries. Challenges are, though, that clearly, even if we go for a normal non-inflamed tonsil in a child that has had a few strep throats, there probably still is some kind of a reason why this particular kid got strep throat and not someone else. And so to what degree these are truly normal is a little bit hard to say. Furthermore, there is, again, an age dependence to developing these diseases. So recruiting into infants and recruiting teenagers, we anticipate a challenge there. Our thymus we get from cardiac surgeries. Advantages, it's an unaffected tissue. Challenges are that children are, again, not completely healthy. We don't really know to what degree and how hypoxia, nutritional differences, and critical illness may affect thymic development. It's hard to imagine that it doesn't have any effect at all. We will need to be careful to exclude some of the genetic causes for heart disease that also affect the immune system. DiGeorge syndrome would be sort of the prime example here. And then we'll have some trouble, again, in filling our age cohorts. That's based both on biology of thymic evolution, where older teenagers just don't have much of a thymus left, as well as the skewed time window of cardiac surgeries, which tends to be skewed towards the younger ages. And again, because of overall less donors, having a gender and racially balanced cohort may be a challenge. And then finally, another very interesting tissue that I'm excited about is lymph fluid. This is not something I think that a lot of places capture, but we had an existing program that is based both on expertise in lymphatic malformations, for which CHOP is a big referral center, as well as cardiothoracic surgeries. And so here we are, again, probably not looking at completely healthy children. We just went through the issues of the kids who have cardiac surgery. And then for the lymphatic malformations, we used to think that these just sort of happen randomly, and it's kind of a collection of otherwise benign lymphatic vessels. And what we've learned in the last few years is that these are actually driven by somatic mutations in signaling pathways, and it's a bit unclear how and if those could affect the cells in the lymph that's present in these lesions. But again, it's very hard to see where else we could get them. The last tissue where we are able to truly recruit healthy children is, of course, blood. And here we're taking advantage of ongoing studies that monitor normal immune development parameters in the microbiome. And this is a very large, both cross-sectional and longitudinal cohort that we'll have access to. Very nicely is that this is a study that is based on a network of participating primary care clinics, which including those that serve underrepresented minority areas within Philadelphia. And so this will truly allow us, at least for this tissue, to have a very diverse and equitable cohort. Some of the challenges here is that because of the many sites, organizing sample collection and shipment can be a challenge. And as we were actually setting up this project, particularly with the link to the microbiome study, we had so many interesting questions and really not enough funds to even ask them all, let alone answer them. So this is just sort of an overview where our samples come from, and you can see that this is really a massive collaborative effort that involves people from oncology, the stem cell lab, immunology, cardiac surgery, ENT, the lymphatics team, ID, allergy, immunology, neonatology. And so we've started collecting our samples. We're quite excited about this. So what are we going to do to them? Oh, sorry, first our age groups. We spent a lot of time trying to come up with something that's both reasonable and financially doable, and we identified these sort of 4 key periods. The first one being children less than 2 months of age, so this is pre-weaning, pre-vaccine, a very immature immune system, a hematopoietic system that really has still has fetal characteristics. Then we have the 2 to 12 months old, which is really sort of your first exposure to antigens. It's when children hopefully get vaccinated and when they're starting to be out and about. Then we have the 1 to 8-year-olds, which is our peak of lymphoid development, but still pre-puberty. And then we have our puberty, where we establish gender-specific differences in hematopoietic and immune parameters. So these are the assays that we will deploy. We've built a custom site-seq panel, as well as an ATAC-seq analysis platform that we will deploy. And whenever possible, we will try to combine this with Codex, and then take advantage of a innovative pipeline built by Pablo Gonzalez Camara at Penn that integrates site-seq and Codex into a spatial transcriptomic map.
Kathrin, 1 minute, please.
Yeah. In addition to this, we will deploy a single-cell nascent transcript RNA-seq platform. So in the last minute, I will share just 2 slides from our efforts on the bone marrow. We have already run 16 total samples, sorted, and built a hematopoietic map. And we've used that to try to answer one of the key questions that has bugged me for a long time, which is, why do babies less than 6 months old do so poorly, but we have so much better luck in curing older children with ALL? And what we found is that when we use this hematopoietic map and we map back blasts from very young infants, we find that most of the blasts are on this arm here, which is B-cell development, but they spill over into other compartments, including stem cells up here, erythroid precursors up here, and myeloid precursors down here. And we don't really see this in the older children. And we also see a crosstalk between stem cells and immune cells. And we also see a steroid resistance signature in some of these very immature compartments that are specific to these young babies, really answering one of the key questions that we've had in the field for decades. And this was just published, so anyone who wants a little bit more detail can go to this paper. So clearly, we need a high-resolution map of pediatric hematopoietic and immune development. Normals for disease stages are needed to be age-matched, and we need to understand age-dependent developmental signatures to understand disease states. I'm not going to read the challenges. We went through them, and I'm looking forward to having a discussion about them with this group. Thank you.
Thank you so much, Kathryn. Please do type your questions to Dr. Byrne in the Q&A function of the Zoom, and she'll try and respond while we have our next speaker present. And our next speaker is Dr. Bartha Maria Knoppers. She's a director of the Center of Genomics and Policy of the Faculty of Medicine at McGill University. She's also chair of the Regulatory and Ethics Working Group at GA4GH and is a member of the International Commission on the Clinical Use of Human Germline Genome Editing. Bartha, I will share your slides. Just
Thank
give me
you.
a second.
Bonjour tout le monde from Quebec. It's great to be on these international conferences. Let me just say I chose my title, Pediatric Research Ethics Growing Up, because the last 20 years or so have seen an incredible increase in the amount and the attention paid to specific issues, the ethics and policy issues concerning children, to the point where you can now almost see what, pardon epidemiologists, a sort of a stratification, and which fits with the developmental aspects of children as well, from newborns and what to do now with whole genome sequencing, to the specific interests of children, to minors, to what are now called mature minors, to finally legally considered to be adults. And in that spectrum, there's quite a wider differentiation in the policies that address the issues in pediatrics. Next slide. So let me just set the stage for my presentation. It's obviously the United Nations Convention on the Rights of the Child, adopted in 1989, and it is the one convention in the world that has every country except one has signed and ratified the convention. And it's the first to recognize children as individuals, as persons, not just members of a family, and the first to recognize internationally that there should be specific attention paid to their needs and appropriate legal protection before as well as after birth. And that's interesting because while they weren't able to include an article on research, they couldn't come to agreement. And while they didn't adopt the specific age, saying now you are an adult, they just said legal maturity as in the different countries. So different countries have different legal ages. They did in Article 24 specifically mention the right of the mother to prenatal care as being part of the interests of the child. Next slide, please. So, no, go back, please. Go back Yes, that one. Okay, next is the consent. No, go back. I saw it. Oh, well, I think I'll just speak to it then in case it's got lost in translation. When you sign and ratify a convention, countries report in, and every 10 years or so, there's a general comment from the United Nations. And their last one paid particular attention not only to the usual issues of consent, assent, confidentiality, and so on, but to the right of the child to have access to confidential counseling and advice without parental or legal guardian consent, particularly with, of course, they were referring to, without mentioning it specifically, to the contraceptive and reproductive interests of minors, and that professionals then should be able to treat or listen to these minors without parental or legal guardian consent. So the slide here is on the whole issue then of the fact that tissue collection is specific for minors, with the parents then providing such consent in the majority of cases, unless you're considered to be a mature minor. Next slide. Next slide. So the United Nations Convention on the Rights of the Child says that the rights or the best interests of the child are a primary consideration and not a sole consideration, because they realize, of course, that children, because of their dependency, cannot be totally autonomous and are dependent, but their best interest should be primary. Next slide. So they also have the right to express their views, depending, of course, on their age and maturity, and the opportunity to be heard in any decisions concerning them. This can go under, medical protection or child protection issues and so on. So they have the right to have their interests be primary, to express their views, and the opportunity to be heard. Next slide. And finally, I think the one of most interest for today's seminar is on the highest attainable standard of health. Now, there's no right to health. It's the highest attainable standard of health, which of course contextualizes it to the particular environment of the child. But it does mention and what the state can offer. And state parties shall strive to ensure that no child is deprived of his or her right to access health services, however and wherever they exist. Next slide. So let's look at some specific and emerging issues, and not the traditional consent, assent, confidentiality. And they relate to return of results, especially now with whole genome sequencing, including incidental findings, and also recontact, because we've seen in the last 5 to 10 years or so, increasingly countries adopting policies saying when the child becomes an adult, however defined in that country, you have to go back and get a new consent. And you're thinking, oh my God, I've got these tissues, I've got this data and so on. What do I do? I don't, and so recontact as part of an emerging right. Next slide, please. So we looked, starting with return of research results, at the best interests and how it's been interpreted in terms of research results. These are research results, not necessarily of clinical significance. But if they are of clinical significance, then the child has a right to their medical benefit from those results during childhood, if they are actionable during childhood. And this is internationally adopted. There are still countries, however, that say no, parents, if they don't want to have these results, can say that they don't want them. However, most pediatricians and pediatric professionals would argue that to respect the child's legal best interests, which are primary, such clinically significant actionable results during childhood should be returned. Next slide. Now, there's difference between Europe and especially the United States on this. Adult onset conditions. What if it's not during the 0 to 18 or 16 or whatever the age of maturity is? Should adult onset conditions not be communicated? And generally, more European protection and Canada and so on would say don't communicate them because it's not clinically actionable during childhood. But now that's being modified to say, unless disclosure prevents serious harm to parents or family members, determined on a case-by-case basis. The child does live in a family, and if those same results could prevent serious harm, then they can or may be returned if so desired. There's also increasing talk, or has been for 20 years, about a right to an open future, which is one reason why these late onset results are still usually not returned. This is not a legal right. It's a right that was came into the literature, sociological, psychological literature in around 1995 and so on, to leave the child the choice at adulthood to decide whether they want to receive such future onset results. Next slide. So then, on top of that, what about incidental findings? Well, these are findings that are not the initial object of research. And again, the literature says that the best interests, again, with significant implications for the health of the minor and effective treatment is available. And in the introduction, Ellen mentioned the Global Alliance for Genomics and Health, and they have just come out after 8 years discussion with a policy proposal for the return of research results and incidental findings that match this statement that you see here. Next slide. Now, there are differences, however, between the European Society, American Society, Canadian College of Medical Geneticists, the American College, and so on. If you look at the European Society, they are more pragmatic. It says, if possible, we contact. There's no duty, so there's no duty to hunt. The decision should be on the best interest. Do what you can with limited resources, and each country should define within its own organizational policy, because there are costs associated with this in universal healthcare systems. Next slide. So in an article we did in Genetics in Medicine, we compare these three statements between the ACMG, American College of Medical Genetics, which has a list of what are serious pathogenic conditions, compared to American Society of Human Genetics, which is for researchers, which seems to be more, create more duties on researchers than you would have on clinicians if you're not in the list. And the European Society, as I mentioned, that was more pragmatic and also raised the issue of resources. Next slide. So what happens? We, in this recontact policy, in a project we're working on, which is a longitudinal developmental project that starts even during pregnancy, what happens if the parent or guardian has chosen to withdraw the minor before adulthood when you're supposed to recontact them? Well, we argued in a paper the yes to the recontact of the minor if the parent had withdrawn. Now, this was very contested. This article took a long time to get through. And we argued no, if it was the minor, him or herself, that was capable of providing assent or mature minor consent, who asked to be withdrawn as an expression of respect for their wishes. Next slide. But what happens if you're in a country that doesn't have a medical maturity age, like Quebec, a province of Quebec, where I am, it says age 14, if a child can go, a minor, to a physician alone without any parental consent for required medical care, and a 14-year-old can participate in research for minimal risk if it's approved by an REB. However, if you're using the mature minor rule, where you decide as a professional who's mature and who isn't on a case-by-case basis, you can't handle this when suddenly somebody has reached supposed adulthood, and so this determination is not possible unless you're actually seeing your participants over time. So you will have to wait till the majority statutory age of medical maturity. Next slide. So just one of my last slides. What do we do about models of consent? What's happening here? Well, if you're really legally protectionist and conservative, you'll say you must get a new consent. And it sounds good. In fact, I think I even wrote on this earlier, about a decade ago, suggesting this. But reality is that it's usually not possible. We live in a highly mobile society. The parents move around, the minor is moved around, and so on. So new consent is one possibility. Another is to simply notify minors at the supposed age of maturity, majority, whatever the country uses. And this is a Canadian Health Measurement Survey where we have a developmental longitudinal study. And we started because a privacy commissioner said you must contact these children at the age of supposed majority, depending on the province. And the results were incredible. If you work with teenagers, you can imagine trying to call anyone before noon. You can imagine the kind of answers you get. Yeah, duh, what? I don't like my parents or whatever. So the attrition was incredible. The impact on the value of the research was such that we asked for permission to notify minors with a letter at their 16th birthday saying that they could opt out and tell us they didn't want to continue, even if it was passive. Obviously, any new samples, you'd have to get a new consent, but passive continuing through linkage of administrative databases. And the most surprising thing is that they loved, the best part was not the research, for getting a real letter. They'd never had a real letter in their life. And finally, of course, you can get waiver of consent by an REB for this recontact obligation. Next slide. And this is my last slide. So in this pivotal Article 24 on the right to the highest attainable state of health, it gives a whole list of indicators and factors that should be included. And in particular, the need to provide children with adequate and appropriate information. And I would think, specific to their interests, I would think in this digital age that, and now with digital schooling in the last 2 years with COVID, that I think the time has come to sort of revisit and revisualize and reformat our approaches to communication with children and minors. So I'll close on that particular note.
Thank you so much, Bartha. Please type your questions in the Q&A section. And with this, I will introduce our last speaker for today. It's Dr. Deanne Taylor. She is the Director of Bioinformatics in the Department of Biomedical and Health Informatics at CHOP. She's studying the genetics and genomics of development and reproduction. And she will introduce dGTEx.
Thank you. So this is a new project that was recently funded by the NIH in a combination of a few different NIH granting organizations, institutes. And I'm going to share my screen. And there we go. So I'll get back to that slide in a minute. Let me bring you to the dGTEx website that was just released. Pardon me for a minute. I'm trying to work through all my that's not what you want. There we go. Sorry, I've got a bunch of things in my desktop here. All right, so the dGTEx website just was released, and it really describes. You can go here, dgtex.org. It describes the surface right now of the project, and I don't have enough time and to really dig down into what the project will be about. But I can tell you that the project is generally going to be sampling tissues. We just heard that there's a lack of normal tissues from children, and this is a project that will be taking samples from children from 4 age groups that will be post-mortem and done at the time of organ donation. Usually, the organ is taken at the time of organ donation. And then we have postnatal, early childhood, prepubertal, and postpubertal age groups. That isn't actually too far from the groups that Kathrin Bernt was just showing.
Maybe, Deanne, maybe maximize the window.
Oh, sorry, it's
perfect. This is good. Yeah.
Yeah, sure. So, so this is still this website will probably change several times in the coming months, but this is the first round. You can click on the project. This will bring you to the NIH website, which I will not do right now, but you can go there and review the project from the NIH point of view. And our overall, the problem that we've had across pediatric research, especially in single cells, is specifically that we do not have normal tissues for children. And we recently had put out, well, a couple of years ago, we put out a paper on the Pediatric Cell Atlas, which Bruce and Kathrin were actually on that paper. And it's an overview of exactly why a pediatric atlas is important for understanding a number of different processes that could actually impact adult health. And also understanding different types of adult processes, including things like regrowth and regeneration of tissues. So the dGTEx project promises to possibly at least produce some data that could go towards all those kinds of concerns. So there'll be 120 donors total. So it's not as big as the original GTEx. There'll be about 30 donors per group, and with hopefully a good mix of ethnicities and sexes in this. And we actually have an LC group as well, which I'll discuss in a minute. So the goal of this is to establish a molecular data analysis resource, as well as a tissue bank, which is an important point. When we harvest the tissues, we're also going to be reserving a lot of that tissue for future studies. So, even though we'll only be doing a minimum, I wouldn't say a minimum amount, but a number of tissues, there's possibilities that different parts of those tissues may also be available to future research that might not be sampled during the initial or the major project. And there's also going to be actively research into having this data act as a reference for neonatal pediatric and adolescent groups, building on the work that's already been done in GTEx. And so the goal is to actually do about 30 tissues total, with about at least 900 tissues per cohort, so about 3,600 tissues total. So the study is going to genotype each individual and characterize all tissues available via RNA sequencing. And right now we're trying to understand from experts and internal participation, what are the best tissues to characterize? What are the most important tissues to characterize? And so that's an ongoing conversation. If you have any particular preferences, you can feel free to email me, and I can bring that, as part of the, we're going to start collecting this from the community, hopefully, what kind of input people have for what tissues would be the best to sample for different age groups. And of course, some of that will be influenced by the types of diseases that are present in children, as well as our need to connect to the original GTEx platform, all the tissues that were harvested and tested in GTEx. And we'll also be doing single cell RNA, open chromatin, and spatial transcriptomic assays across multiple tissues. And there's also developmental specific genetic effects on gene expression. So there's going to be an eQTL style or QTL style analysis being planned for this, just like in the original GTEx study. So molecular profiling, we're going to be taking blood from each donor with whole genome sequencing, which will obviously be protected data as like the original GTEx. Every sample tissue per donor will be sequenced with bulk RNA-seq, and then a subset of these samples selected for quality and basically for quality on multiple different fronts are going to be assessed for whether or not it goes into single cell and spatial multi-omic profiling. Now, one of the things that we're doing at CHOP, which I'll discuss in a minute, every tissue sample is going to have a pathology review performing histological images of those tissues. And those tissues are, and the pathologists that are going to be selecting, that have been selected to, or that have been participating in this project, come from multiple pediatric centers. So we have pediatric pathologists that are going to be specifically looking at the tissues that they're very familiar with and producing a report, both an image and an overall report of the tissue itself. And all collected tissue samples are going to be deposited in a biobank for future studies. So these are the minimal number of tissues that will be sampled. And we have two different groups doing brain and non-brain. So we're separating it out into brain and non-brain tissues. But, whole genome sequencing, bulk RNA-seq, one per tissue, and single cell and spatial multi-omics, we have not established that yet. Definitely histology and biobank information will be stored for each tissue. So our timeline is we are now in the process of jointly developing protocols and LC research, basically protocols of both LC and molecular biology and bioinformatics and informatics work is being done right now. And then we're partnering with NDRI, and that will be, we're expecting to start getting samples sometime at the end of this year. So tissue collections will start. And then data will start being produced next year and be hopefully produced and released, and data releases starting in year 2 after tissues have been collected. And there'll be rolling and scheduled releases, probably much like GTEx has done, although I can't speak to that right now. And funding has been, these are just 2 of the institutes. There's 2 more, but it's the National Human Genome Research Institute and ICHD are part of this, and some other institutes as well, and this will be updated. Our structural organization, we have an LC group as part of the BPC. So I'm going to move over here really quickly to my PowerPoints to show you a little bit about the overall structure schematic. These are slides from Kristen Ardley, who's one of the PIs of the LDAC. I'm one of several PIs in the BPC, which is the tissue sample collection arm, where we have our group of tissue donors. And then the LDAC will be doing an experimental plan. This is the Broad Institute in Yale, and the BPC is a consortium between the University of Maryland, CHOP, and the NDRI, which is the national donor. I'm sorry, I'm lousy sometimes with these. Then I think it's the National Donor Registry, but their group is providing the OPOs, the organ procurement organizations. They're organizing all the organ procurement organizations, and the data is coming into CHOP for biobanking, pathology review, and informatics gathering. And then that data and tissues are being sent to the LDAC, who are doing all the work on single cell and molecular characterization of the tissues. And so, just as these are the people in the LDAC teams. Kristen Ardley and Ninad Sestan are the PIs of the LDAC, and these are all the other people contributing to the project. And then at dGTEx, the NDRI, CHOP, and University of Maryland, Tom Blanchard is doing the brain. And at Yale, Ninad's group is doing the brain. So they're doing the brain sequencing and analysis. And Tom Blanchard's group at University of Maryland is doing the brain procurement, histological characterization, and preparation. And our hospital teams that are working with CHOP, and these are the groups that are working with us as part of the work on the pathology. And that is pretty much it for me. It's not a very organized presentation, but we're still very early on in our assembly. And so this is kind of a at the minute information session for about dGTEx. And if you have any questions for me, you can certainly email them to me, and I'll be happy to connect you to anyone that might be able to answer your question if I can.
Thank you so much, Deanne. So this leaves us about 7 minutes for additional questions or to revisit any questions that you consider extremely important from the Q&A section of the Zoom. Deanne, if I could ask you to lead the following 7 minutes, and Jenny Root will help us with the questions, posting them in the panelist link, if there are any.
So,
Bartha, you have a question? I think your hand is raised.
Yes, I have a question. Actually, it's a comment. I tried to put in the Q&A, but I must be technologically challenged. I forgot to mention that the GH, the Global Alliance for Genomics now actually has a whole section of topology of consent clauses for genomic studies in pediatrics coming from around the world that we've put together to help researchers at around grade 6 language. Hopefully you'll find something that you can use there. And also, most importantly, I forgot to mention the HCA itself has an ethics and governance working group. And this year, our concentration, because of the pediatric work that's just been awarded, is on all the pediatric ethics issues. So if you have specific pediatric ethics issues that you'd like the ethics and working group of the HCA to work on, just contact me and we'll make sure we'll have a look at it and try to help. Thank you.
So we have I'm wondering, Dr. Byrne or others, Katherine, people are wondering, I think Jenny Root asked if you can comment on the challenging of stratifying samples into biologically relevant age groups. Will it be important that age categories be consistent or at least relatable across different organs for eventual integration, especially in the case of immune cells, where there'll be resident cells captured in many organs? Would love to hear what you think would be the most impactful way to bring together work that's being done in different groups.
Yeah, I completely agree. This is a huge issue, and we actually had months of conversations and almost fights even within our group. And what we initially proposed to the Hans-Zuckerberg Foundation was a lot more brackets than we finally settled on. It just ended up being that they said we can finance this. So yeah, I don't have a good answer. I mean, ideally, what we would have, of course, loved to do is get get all the tissues from the same donor and have much more fine sliced age groups, and that's just not feasible. What we will do is that we just capture age very detailed and publish it alongside our HCA data, in the hopes that it will be useful and maybe can be complemented or supplemented by other people's work. But yes, absolutely, it's going to be a challenge.
And another challenge for us is also determining when puberty starts. That's all going to be annotation that we have to include. So even though there's age separations and how we are going to collect data, it doesn't, those are kind of category for us, they're buckets, but they're not necessarily, I mean, people can obviously, we don't necessarily have to group ourselves, for example, by age in those buckets. We really can be grouping them by what we consider important markers, and so can the people using the data. So it is an open question for age groups, but I think this is just really a convenient bucket for us, at least in the dGTEx. And there's a question, how do you determine what omics are most important to include in your atlas? I guess that could go across a lot of different people. I'll just say that in the omics, which are most important to include, are always a scientific question. I think for dGTEx, it's really the most tried and true genomics methods that people feel secure and comfortable with, that they're not going to change massively in the next couple of years. Although, of course, with single cell, that's not always a guarantee. But things like ATAC-seq and single nuclei sequencing, because a lot of this is frozen tissue, we have to reflex to single nuclei. But I know Katherine or Donna, maybe you could answer that. What's the most, and how do you decide what are the most important omics to include in any atlas you design?
Yeah, that's a great question. Obviously we want to look at, like right now we're looking at gene expression, but ultimately I'm looking at tissue. Protein is going to be really important in establishing proteomics. So really, it's not just what's the gene activity, but also how is that translating into what's actually happening in the tissue, and is it happening differently in different areas? So we're really interested in that sort of spatial genomics, spatial proteomics, and trying to link that together, which is an overwhelming undertaking. I'm not sure how we're going to get this done, and then I still have to operate. So
Yeah, and from our perspective, I think we did not actually think very much about cross other atlases integration. For us, it was more, what are we thinking are the best platforms to use, and particularly within the hematopoietic system that just has so many different cell types, and they're all sort of just hanging out in there. Looking at them transcriptionally, and particularly with this sort of almost a run-on type sequencing with a metabolically labeled RNA-seq was exciting for us. So at the same time, realizing that that's probably not something that anyone else will be doing for any other tissue. So again, I don't think I have a good answer. We've at least tried to stay with one of the commonly available site-seq panels, although it has a lot of antibodies that we don't really care about, and then just add a few in when we designed our site-seq panel. So it's like the BioLegend B panel. So at least that hopefully will be the same across multiple different atlases, and that was a choice by design and for that reason. But beyond that, not really something we have considered. And I agree, though, it's a very important question, because ideally, if everyone could use platforms that are widely available, that would make it easier to supplement. You know, maybe Donna is capturing some age groups that we're lacking from her bones, right? What an opportunity.
Yeah, we throw away a lot of bone marrow.
I was
I know, every-
We should
every
talk.
thinking you and Donna would be a great pair to talk.
Yeah.
Because there's so much available. You both overlap in a lot of different ways.
Yeah.
Yeah.
Yeah,
Absolutely.
I agree.
Well, unfortunately, we're drawing to the end of the seminar. I want to thank everyone who joined today. I want to thank our wonderful speakers and panelists. We hope to see you at our next seminars in the series. We want to thank Chan Zuckerberg Initiative for generous support of this series. And please do join the HCA network and email the participants if you have any additional questions. Thanks, everyone, and have a great day.